Hello, my name is Mark Velichko, and I would like to present my project called The Use of Mycelium Bio-Composites for Environmentally Friendly Insulation. The production of traditional insulation materials consumes significant amounts of energy, polluting the environment. Therefore, the requirements for sustainable construction demand for new, low-cost and environmentally friendly materials. One of such materials could be fungal mycelium, which combines with organic matter from agricultural and industrial waste to form a biocomposite material with unique structural properties. When thinking about fungi, for many people the first thing that comes to mind is the image of a mushroom. However, mushrooms are only the fruit bodies of a fungus, which produce spores in order to reproduce, much like apples on a tree. The actual tree, the vegetative part of the fungus, which creates the fruit bodies, is called mycelium. Mycelium is usually invisible, buried in its substrate, and consists of a dense mat of branching, thread-like filaments called hyphae, through which nutrients, cytoplasm, and genetic information travel. One of the most important properties of mycelium is that it acts as an adhesive agent, binding its substrate together to form a dense block. It has also been found to be waterproof, fireproof, and contain antimicrobial properties. The mycelium is very easy to produce, as it can colonize a substrate of organic matter in a matter of days without requiring much human input. Not only would the production of mycelium insulation be cheaper and easier, requiring less manpower and practically no complex machinery, it would also be environmentally friendly, containing no toxic chemicals, and break down easily once it is no longer needed. An additional benefit of mycelium composites is an opportunity to use the fungus for both insulation production and mushroom cultivation for food. The aims of this investigation are to test the effectiveness of mycelium insulation and compare it to polystyrene, a conventional insulation material. In order to successfully produce many mycelial bricks, it is important to develop a method to grow the fungi efficiently and in the correct quantities. The mycelium used in this investigation was grown by mixing 300 grams of fungal spawn with 500 grams of hydrated oak pellets, which had been pasteurized in boiling water for 1 hour. After 10 days, the fungus began fruiting, signaling that colonization was complete. After the colonization was complete, the mycelium composite was baked for 1 hour at 90 degrees Celsius. The baking was essential to stop further growth and remove moisture, leaving behind a firm product. After baking, the mycelium block was cut up into slices 2 centimeters thick, ready to be glued together using PVA to form a container. 3 experiments were designed in order to compare the performance of 2 types of mycelia and 2 types of polystyrene. In the first experiment, a light bulb was placed inside the container, and the time taken for the temperature of the air inside the material containers to reach 40 degrees Celsius was recorded. The air inside both mycelium samples took much longer to heat up than in the polystyrene, which can be explained by the greater thermal capacity or heat absorption properties of mycelium. In the second experiment, the inside air was heated up to 3 different temperatures until a steady state was reached, and the supplied electric power needed to maintain the constant temperature was measured. This was done to estimate the thermal conductivity, which is an important characteristic of insulation materials, and measures the ease with which heat can travel through a material. According to the results, the thermal conductivity of mycelium is slightly higher than polystyrene. In the last experiment, the time taken for the air inside the container to cool down to 27 degrees Celsius was recorded. The results showed that the air inside mycelium samples took much longer to cool down, ensuring the delay in heat transfer through the walls. This effect is called thermal buffering and might be particularly useful for countries with a hot climate, making it possible to control and prevent overheating of a building. The results of this investigation show that mycelium insulation would be particularly useful in developing countries in Africa and the Middle East because of its low production costs, environmental sustainability, insulation properties, and an additional potential for mushroom cultivation during the mycelium growth. Moreover, the mycelium's excellent thermal buffering properties ensure time delay for the midday peak temperatures to penetrate the wall to the interior of the building. In the future, it would be interesting to investigate different factors that affect the insulative properties of a fungus, such as different substrates and growth conditions. Studying this in more detail will enable us to optimize this new type of insulation.